Okay, our next podcast continues with our look at the macromolecules. We've discussed the first two in the previous podcast on carbohydrates and lipids. The second um, two include the proteins and the nucleic acids. So again, these are the large macromolecules that play important structural and functional roles in living things. So proteins is what we're going to look at. They have many different structures and many different functions as well. They can be long, very long chains of amino acids. They are polymers. So we mentioned that polymers are repeating subunits of monomers, and the monomers are um, amino acids, and so that is what they are made up of. And they have a lot of different roles or functions in the body. Here's an example of various types of proteins and their functions and some examples of those. So a lot of structural proteins are found in the body to give support, like these examples of connective tissues in human body, the hair and horns, feathers, skin appendages, things like that are made up of proteins. Also, enzymes, we'll look at enzymes in a minute, storage proteins, transport proteins, hormones are proteins. So, and then we have defense proteins. So there's a lot of different functions that proteins have in our body. We'll look at enzymes here. Enzymes are a type of protein that act as a catalyst. That means that they speed up a chemical reaction. So here's an example. This big purple thing here is an example of our enzyme. The enzyme is sucrase, and when it is in contact with a certain substrate, the substrate being sucrose, it can speed up the reactions to separate this into two separate molecules to break it down. In this case, we're talking about a dehydration process here, but there's a lot of other processes that enzymes help. So they're important in speeding up chemical reactions, like in metabolism and that type of thing. So enzymes are important. So again, the proteins are made up of repeating subunits of amino acids. Here are a list of those amino acids. So there's 20 of them. There's 20 amino acids, and so there's different sequences of these amino acids in the different bases. So amino acids differ in their properties due to different types of side chains, and but the carbons and oxygens are the same, but we have different side chains that are in these amino acids, and so we'll look at that. But amino acid polymers is what a protein is. Now these can be basically they can be just simple small structures, the amino acids that is, the proteins that is, with just a few amino acids, or they can be very large structures with lots and lots of amino acids taking on elaborate shapes. And so they can have coils, they can have folds, they can have twists. Here's a couple of different models to kind of show or describe some of these features that these proteins have. Because with all the coiling and the folding, they take on unique shapes, and their shape is very important in relation to their function. Here we see a groove here that's like something could fit right in there. So back up here at our example of our enzyme. So this is representing part of the shape of the protein right here, and that substrate fits right in there. If it didn't have that shape, it would not fit right in there. So these are just different ways of representing that. This is called a ribbon model. This is called a space filling model. And so they can get very large. Here's another computer animation. We looked at one of these, one of our other podcasts. But these would show, shows a antibody protein that we produce in our body to fight off a virus here, a flu virus. And you see the fit of it right here. It fits right exactly in there, like a key. And so that fits very important. So because these can get large, we have different levels of describing these proteins. It starts with just a simple explanation. That's called a primary structure, where we just simply list the amino acids. And then we go to more complex, called a secondary structure. This is where we look at those coils and folds. And then a tertiary is kind of like a three-dimensional group. We look at a view. We look at how they kind of fit in all their folds and fit together in the chains. And then a quaternary structure is when you have multiple chains of amino acids that end up hooking together. So that's in your complex proteins there. So here's an example of a primary structure where we just simply list the amino acids out here in a straight list. So there's 25 amino acids in this example. And then secondary is going to look at the folds, how it actually, the physical fold of the molecules when they come together, if there's folds or spirals or whatever. And then tertiary and quaternary are for your very large proteins, where how the folds come together in making up the whole polypeptide. And then quaternary is when we have multiple chains that hook together for large proteins. Let's see, I think we have, no, so we had an example of one. So certain fibers are made up of proteins that are twisted together to make multiple, to make a quaternary structure there. Okay, so that's our proteins, and then our last macromolecule that we're going to look at is our nucleic acids. And the nucleic acids are important structures. There's only two types of nucleic acids. We have, in this diagram, we have DNA and we have RNA. DNA has a sugar called deoxyribose, nucleic acid, and then RNA has ribose, nucleic acid. So those are two nucleic acids. The nucleic acid's main role in living organism is to carry genetic information. So we have a whole unit devoted to genetics, and we'll talk a lot about it. Here we're just kind of introducing the molecule itself, where the genes are found, which has the code for our traits. So the DNA is found in our nucleus. It's made up of two strands. Now your book always represents by using this blue ribbon model here. And you see there's two of them, because DNA is made up of two strands. And so we call it a double, and this spiral shape is referred to as a helix. So we call this a double helix. And RNA is just a single ribbon, and it is not in a helical shape. But these are our two nucleic acids. Now they are, some of their role is DNA carries information, and then RNA copies it from DNA, and then it will take it out of the nucleus. So this is our nucleus of our cell right here. takes it out of our nucleus and then into a protein where the information is read, or into a ribosome where the information is read and then made into a protein. So that's the ultimate goal of our DNA, our genetic code, is to make proteins. And so we'll learn all about that. We get into that process in depth in genetics. So we'll learn about that more in depth in genetics. But that's the role of the nucleic acids. So nucleic acids are made up of repeating subunits. They are a polymer, and the repeating subunit is called a nucleotide. So this shows one nucleotide right here. It has 3 parts to it. It has a phosphate group, so we learned about that functional group. It has a sugar right here, and it has a base attached to it. All right, and so the bases are the only thing that differs. So we have a whole bunch of these nucleotides, and the only thing that differs in them is the bases. There's 4 different bases in DNA: C, T, A, and G. Stands for cytosine, thymine, adenine, and guanine. And so those different letters represent the alphabet, genetic alphabet that makes up the codes. And so those codes are going to produce various traits. All the traits you have, the traits we see in all plants and animals, come from that code. RNA is the only thing different in RNA is that it's a different sugar, but also it has one difference right here. It doesn't have thymine in it. It has uracil in place of thymine. Otherwise, it has all of the same bases in the nucleotide. Okay. Now, well, there's 2 groups of these. The first 2 are called pyrimidines, and they pair up with the purines, which are the adenine and guanine. T always pairs with A, and C always pairs with G. We'll learn more about that pairing a little bit later when we get into genetics and we see how the DNA copies itself. And so this is an example of a DNA that's separating, and you can see that pairing right here. Right across, the C is a G, and right across always the A is a T. And so this holds the 2 strands together. This is showing one that's unzipping and unwinding right here and forming 2 new strands, or forming 1 new strand. All right. The last thing we can look at here is this is a review. It's at the end of your unit here, and it kind of the chapter, and it reviews all these large biological molecules. Good figure here: carbohydrates, lipids, proteins, and nucleic acids. Shows you examples of each one of these and the functions of each one of these. And that is where we end for this topic. And so we will pick up in the next podcast with our next set of material.